Usually, when we think about the anatomy of the brain, we picture this just vast, tangled forest of wires,
Right, like a bowl of microscopic spaghetti.
Exactly, just billions of neurons shooting electrical sparks back and forth. And it creates this expectation of, well, pure structural chaos. But if you zoom in, like really step into the world of nanoscale structural biology, that chaos completely vanishes.
It really does. It becomes incredibly ordered.
Yeah, we're actually looking at a landscape that is just mind-bendingly precise. So today, for our deep dive, we are exploring a hidden microscopic scaffolding called the membrane-associated periodic skeleton, or the MPS.
Which is such a fascinating structure.
It is. We're cracking open a massive 2022 Nature Communications paper by Robozio, Xiaowei and their team. And our mission today is to figure out how this newly discovered nanoscale slinky basically dictates the physical size of your nerves and, well, how your brain actually wires itself together.
And that's pretty wild when you consider that this structure is just so foundational to how we think and feel, right? But it's so unimaginably small that it entirely evaded detection until just a few years ago.
Wait, really? Just a few years ago?
Yeah, I mean, in neuroscience, we've historically focused on the electrical sparks, the neurotransmitters jumping across synapses. We completely overlooked the physical pikes holding the whole system open and giving it shape.
So to visualize this, let's give you listening a clear image to sort of anchor all this highly technical biology. Imagine a rope ladder.
Okay, a rope ladder. I like that.
But instead of hanging flat against the wall, this rope ladder is twisted into a long, hollow cylinder, like a tube. And the solid wooden rungs of that ladder, they're made of a protein called actin.
Right.
And then the flexible ropes running down the sides, connecting those rungs, are made of a protein called spectrin.
That is actually a highly accurate mental model. The actin forms these continuous ring-like hoops, and the spectrin forms tetramers, which are basically complexes of 4 individual molecules.
Oh, so they act like spacer springs between the rings.
Exactly. They act as flexible spacer springs between them, and the spacing between those actin rungs is utterly rigid. It is exactly 190 nanometers every single time.
Wow. 190 nanometers.
Yep. It creates a one-dimensional periodic structure running just beneath the outer skin of every long nerve fiber.
Wait, I feel like I've heard of actin and spectrin skeletons in other types of cells before, like red blood cells, right? Don't they have a similar skeleton to help them squeeze through tight blood vessels without popping?
They do, yeah, the erythrocytes, but the internal geometry there is completely different.
How so?
Well, red blood cells use actin and spectrin to form a 2-dimensional polygonal lattice, so it looks more like a microscopic fishing net spread across the entire inside of the cell membrane.
Oh, okay, so a net versus a tube.
Exactly. What we are looking at in neurons is specifically a one-dimensional tubular structure. Plus, the molecular building blocks are distinct. Red blood cells rely on a specific variant called alpha-I spectrin.
And the brain uses something else.
Right. In your brain, the neurons are using alpha-2 spectrin paired with beta-2, beta-3, or beta-4 spectrin subunits. So it is a highly customized toolkit specifically evolved to support long neural cables.
Okay, let me do a quick recap here so we don't lose anyone. You've got this microscopic tube holding open your nerve fibers. Actin hoops make the rungs, spectrin makes the ropes, and it's all spaced exactly at 190 nanometers.
You've got it.
But wait. Actin is notoriously unstable, isn't it? From what I remember from cellular biology, actin filaments are always trying to do this thing called treadmilling. Yes.
The treadmilling problem.
Right. They have this natural chemical tendency to constantly add new building blocks to one end, the fast-growing end, while simultaneously shedding blocks from the slow-growing end. So if these actin rings are just continuously growing and shrinking on their own, how on earth does the neuron maintain a perfectly rigid ladder without it just falling apart?
This is where Zhu and Zhuang's team made some of their first incredible discoveries. They found out how the cell locks the structure down.
What do they do?
Well, they found that the neuron physically caps both ends of every actin filament in the ring. They identified a protein called adenosine that acts like a literal plug on the fast-growing end of the actin.
Okay, so that stops it from getting bigger.
Right. And another protein, tropomodulin, caps the slow-growing end. By plugging both ends simultaneously, the cell stops the treadmilling process cold.
Wow. So it freezes the actin ring in place?
Exactly. Its diameter cannot grow out of control or spontaneously disintegrate.
Okay, so the ladder is capped and stable.
Yeah.
But a basic frame doesn't do much on its own, right? For it to control brain wiring, other molecular machines must be attaching to it.
Precisely. The researchers wanted to uncover the full interactome.
The interactome, meaning all the interacting proteins.
Yeah, every single protein that physically docks onto this 190 nanometer scaffolding. To do that, they took cultured mouse hippocampal neurons, as well as entire adult mouse brains, and used a technique called co-immunoprecipitation, or co-IP.
I love the mechanics of co-IP. It's basically molecular fishing. Imagine you listening want to know what fish naturally swim together in a kelp forest.
That's a great analogy.
You take a very specific piece of bait, stick it on a hook, and see what you drag up from the depths. In this experiment, the researchers used tiny magnetic beads coated with highly specific antibodies as their hooks.
And for bait, they used known parts of our rope ladder, the beta-T spectrin, the alpha-T spectrin, and that Deucin cap we just talked about.
So they basically dropped those magnetic beads into a neuron lysate, which is basically a soup of completely liquefied brain cells.
Right. And the antibodies grab onto the spectrin bait, and crucially, they drag along any other protein physically tangled up with or bonded to it.
And then they just pull them out.
Yep, they pull the beads out with a magnet, wash off all the extra liquid, and then run the catch through a mass spectrometer to identify exactly what they got.
Which is just a brilliant piece of machinery. A mass spec essentially shatters the trapped proteins into tiny fragments using electrons, and then weighs those fragments using magnetic fields.
It's like finding a chemical fingerprint.
Exactly. Because every single protein has a unique molecular weight. And their catch was staggering, wasn't it? They found 480 candidate proteins docked to the structure in cultured neurons and 670 proteins in the whole brain samples.
It completely illuminated the hidden, bustling city inside the axon, but finding them was really only half the battle.
Right, because just because a protein gets dragged up in a magnetic net doesn't mean it's actually part of that perfectly spaced ladder. It could just be a cellular bystander that got sticky during the liquefaction process.
A very critical distinction. Co-IP only proves two things are sticky. It doesn't prove structural geometry. To verify that these proteins were actually spaced at perfect 190 nanometer intervals, they had to visually see them.
But you can't just look through a regular light microscope.
Yeah.
Because of the diffraction limit of light.
Exactly. Anything smaller than about 250 nanometers just smears together into a blurry blob because of how light waves interfere with each other.
So a 190 nanometer gap is essentially invisible to standard optics.
Right. So to bypass this entirely, they use STORM, stochastic optical reconstruction microscopy.
STORM. I always visualize STORM like trying to see the individual light bulbs on a distant Christmas tree. If the whole tree is plugged in, it's just a glowing green triangle.
Oh, that makes sense.
But if you have a chemical trick to make the bulbs blink on and off randomly, just one or two at a time, you can photograph each individual isolated flash. You take tens of thousands of pictures, pinpoint the exact mathematical center of each light pool, and computationally reconstruct the entire tree with nanoscale precision.
That is exactly what STORM does with fluorescently tagged molecules inside the cell. And when they mapped their caught proteins with STORM, the blurry smears resolved into gorgeous, crisp, periodic stripes.
Wow, so they saw the actual ladder?
They did. They confirmed that those capping proteins, along with other structural stabilizers like dematin and chronin 2b, were perfectly distributed along that exact grid. But they also wanted to push the system to its breaking point.
What do you mean?
They wanted to know what happens if the ropes of the ladder are chemically faulty. So, beta-T spectrin has two critical ends. One is the CH domain, which physically grabs the actin rungs. The other is the pH domain, which anchors to the lipid membrane on the inner surface of the cell.
Okay, so the spectrin rope is reaching inward to grab the actin rung with one hand and reaching outward to grab the outer skin of the cell with the other hand.
Exactly that mechanism. So they genetically engineered neurons to produce mutant spectrin missing either that CH actin-grabbing hand or the pH membrane-grabbing hand.
And what happened?
When either domain was deleted, the entire 190 nanometer periodic distribution vanished. The ladder completely collapsed under its own lack of tension.
That is wild. Let me just synthesize this for a second because it's a huge shift in how we picture a neuron.
Go for it.
So we've got our actin rungs capped so they don't treadmill, held exactly 190 nanometers apart by spectrin ropes. And those spectrin ropes must actively hold onto the cell's outer membrane to keep the whole hollow tube open.
Right.
If any anchor fails, the bridge collapses. But this microscopic tube isn't just a static piece of PVC plumbing, is it? Because that brings us to the most surprising catch from their magnetic fishing trip, non-muscle myosin II, or NMII.
Finding myosin here forces a complete paradigm shift. I mean, we normally associate myosin motor proteins with our bicep or heart muscles, where thick filaments of myosin actively slide past actin to generate force and visible contraction.
Right, muscle flexing.
Exactly. So finding a tension-generating motor inside a supposedly passive nerve cable is deeply unexpected.
The physical geometry of how this motor fits into the ladder is brilliant.
Yeah.
NMII forms these bipolar filaments. Think of a microscopic rod about 300 nanometers long. The N-terminus, which is the front end, has these tiny motor heads that actively bind to actin and do the physical pulling. The C-terminus, the back end, is essentially a long anchor tail.
So the researchers needed to know how this motor was positioned within the scaffolding. Was it sitting vertically, stretching between two different actin rungs, acting like a vertical spring to pull the rings closer together?
That would make sense.
It would. But to find out, they used two-color storm imaging. They painted the N-terminus motor heads red and the C-terminus tails green.
This is where the visualization is so clever. Because if the myosin was stretched vertically between two different rungs, which we know are 190 nanometers apart, you'd see a red flash and then a green flash further down the ladder.
Exactly, but that's not what happened at all.
What did they see?
The storm images showed both the red motor heads and the green tails perfectly overlaid together, localized right in the center, midway between the vertical spectrin ropes.
Wait, so they weren't stretching between rings?
Nope. The NMII filaments weren't stretching between different rings. They were sitting horizontally, entirely inside individual actin rings.
Inside the rings?
Mm-hmm.
Which means the motor isn't pulling the ladder rungs vertically closer together. It is hugging the inside of the ring and pulling it tighter circumferentially.
Yes. It radially squeezes the actin rings. The cytoskeleton is acting like an active cellular corset, dynamically controlling the exact diameter of the nerve axon.
To prove this corset was actively squeezing, they had to cut the laces, right?
And it did. They used a chemical compound called blebistatin.
Blebistatin.
Yeah, it essentially paralyzes the myosin motor heads. They can no longer grab the actin to pull. And to double-check their work, they also used hhRNA.
Which is a genetic technique, right? Where you introduce customized pieces of RNA into the cell that physically intercept the neuron's genetic blueprints.
Exactly. So the cell literally stops manufacturing the NMIM motor proteins altogether.
And the results?
The results of both paralyzing and genetically deleting the myosin were functionally identical. The vertical spacing of the ladder didn't change at all. The tube didn't stretch out lengthwise. But the diameter of the axon increased by 20 to 30%.
Wow.
Without the motor's radial tension, the corset just popped wide open.
Okay, we really need to pause here and recap, because a 20 to 30% increase in a nerve's physical diameter is a massive deal for brain function. Let's think about a fundamental concept in neurobiology called cable theory.
Oh, cable theory, absolutely.
So the axon is basically an electrical wire transmitting data. In physics, if you increase the physical diameter of a wire, you significantly decrease the internal electrical resistance. The wider the wire, the faster the electrical signal, the action potential can travel down it.
That's spot on.
I like to picture a blood pressure cuff wrapped tightly around a soft garden hose. If you rip that cuff off, suddenly the hose expands, the resistance drops, and the volume and speed of the water rushing through it changes completely.
By squeezing or relaxing these microscopic actin rings, your brain has a built-in mechanical mechanism to tune how fast a specific nerve circuit processes information.
That is just incredible.
It completely rewrites our understanding of the axon. It is not a passive biological copper wire just blindly conducting sparks. It is a dynamically tuned conduit, constantly adjusting its own internal resistance and transmission speed based on myosin tension.
So this internal corset fundamentally controls the speed of the neuron's internal signals. But neurons don't exist in a vacuum, do they? They have to physically talk to each other to form complex circuits.
Right.
So how does the scaffolding inside the cell affect what's going on outside the cell?
Well, the researchers didn't just pull up internal cables and motors from their mass spectrometry run. They pulled up transmembrane proteins.
Transmembrane meaning they cross the membrane.
Right. These are large proteins that pierce right through the outer lipid skin of the cell, interacting with the outside world. They found ion channels like Kv1.2, which help regulate the electrical firing we just discussed. They found complex signaling molecules. And critically, they found cell adhesion molecules, or CAMs. Specifically, proteins designated as L1CM, NCAM1, and CHL1.
Cell adhesion molecules are basically molecular Velcro. They stick out into the extracellular space, blindly feeling around for other nerve cells to grab onto.
Exactly.
And this is vital for a biological process called fasciculation. In a brain as complex as ours, individual microscopic axons need to bundle together to form cohesive, macro-scale neural tracks. It's like taking thousands of fragile fiber optic cables and wrapping them into one major, highly protected trunk line.
But this molecular Velcro isn't just haphazardly glued all over the outer surface of the cell. Because these cams are anchored directly to our 190 nanometer rope ladder on the inside of the cell, the Velcro hooks protruding on the outside are perfectly spaced at 190 nanometer intervals.
Oh, I see where this is going.
Yeah, here is where the nanoscale geometry translates directly into macroscale wiring. When two individual axons physically touch each other, their internal MPS structures naturally align perfectly in phase.
Meaning the actin rung of cell A lines up exactly with the actin rung of cell B.
You've got it.
Wait, I'm struggling with this part, actually. Let's think about this logically from the perspective of the cell. If the neuron already has all this sticky Velcro floating around on its outer surface, why does it matter what the skeleton inside is doing?
What do you mean?
Well, shouldn't sticky molecules just naturally stick to other sticky molecules when two cells bump into each other in the brain? Why is an internal rigid spacing a prerequisite for them to stick?
It's an excellent question, and it comes down to understanding the fluid mosaic model of the cell membrane. The cell membrane isn't solid. It's made of liquid lipids. It is effectively a microscopic coat of oil.
Oh, right. It's fluid.
Yes. So if a surface protein isn't securely anchored to a hard structure inside the cell, it just floats and slides aimlessly in that oil.
Ah, I see.
To test if this free-floating Velcro could work on its own, researchers used that HHRNA technique again to knock down the beta-tie spectrin, destroying the internal slinky entirely.
They broke the ladder again.
Right. They also ran a trial knocking down a protein called anchorin B, which acts as the specific molecular bolt that physically pins the adhesion molecules to the spectrin skeleton.
And when the internal skeleton was destroyed, but the cell still had all its molecular Velcro floating on its surface, what happened?
The axons just slid past each other. They completely failed to bundle into tracks. Furthermore, the density of synapses, the actual physical communication junctions where memory and learning physically occur, plummeted.
Wow. So it decisively proves that chemical presence on the membrane is entirely useless without physical organization beneath it.
Exactly. If those adhesion molecules are just slipping and sliding freely in the liquid lipid membrane, they can never form a stable, load-bearing connection with another cell.
It's like a complex jigsaw puzzle. You could have all the right pieces in your hand, but if you throw a handful of loose pieces at another handful of loose pieces, they won't spontaneously interlock.
Right. They need to be held rigidly in a fixed frame so their complex edges are forced to catch and hold. The MPS provides that rigid 190 nanometer frame for the brain.
This is just mind-blowing. So if we step back and look at the massive picture we've built today for you listening, we started with these microscopic actin runs, perfectly capped to stop their natural treadmilling, held exactly 190 nanometers apart by spectrin ropes.
Yep, the basic frame.
Then we saw how mass spectrometry fishing and super-resolution storm microscopy revealed non-muscle myosin motors sitting entirely inside those rings, constantly radially squeezing them like dynamic corsets to dial in the electrical resistance and speed of our thoughts.
The tuning mechanism.
And finally, we saw how this entire internal tubular ladder acts as a precise geometric pegboard, anchoring molecular Velcro in perfect alignment so our neurons can physically stick together and form lasting memory circuits without slipping away in the liquid membrane.
It really is a breathtaking masterpiece of structural biology and evolutionary engineering. But, it leaves us with an incredibly provocative and somewhat fragile reality.
How so?
Well, this structure is highly dynamic. It relies on the continuous active tension of myosin motors and the flawless geometric alignment of highly fragile spectrin springs. So what happens when that nanoscale geometry is violently disrupted?
Like in a traumatic brain injury.
Exactly. Think about traumatic brain injuries, the sheer physical shockwave of a concussion rippling through the delicate brain tissue. Or consider the slow, steady degradation of cellular proteins that inevitably happens as we age.
Oh, wow.
If this 190 nanometer periodicity is the the absolute physical prerequisite for our neural circuits to stay bundled together and fire at the correct speeds, could the very beginning of cognitive decline, or the literal unraveling of a cherished memory, start precisely here?
The microscopic snapping of a single overtension spectrin rope. That is an incredibly haunting yet profound image to leave on. The idea that everything we are, our complex memories, our behavioral quirks, our ability to learn and adapt, is fundamentally physically dependent on a submicroscopic slinky holding its rigid shape just under the surface.
It definitely changes how you look at the brain.
It really does. I highly encourage you listening to think about that hidden internal architecture the next time you sit down to learn a new skill, or even just move your hand to pick up a coffee cup. You are relying on billions of perfectly spaced actin rings squeezing and relaxing in perfect harmony to make that simple action happen.
Billions of them, all working together.
Thank you so much for taking this deep dive with us today into the precision engineering of your own mind. Catch you next time.